Okay, in the second part of our Capybara tutorial, Wenjin's going to be walking through the GitHub tutorial. Yeah, over to you, Wenjin.
Okay. Can you guys all see my screen? Okay. So personally, I believe we have a pretty detailed GitHub page for instructing through how to run through Capybara. So I will do a step-by-step walkthrough and I hope it will clarify something that might be confusing in this tutorial as well. Feel free to jump in anytime and let me know if anything is not clear. So as I described before, we do, in the first step, we perform a tissue level classification. So the bulk transcriptome profile are mined from the platform ARCHS4, and it contains most of the published RNA-seq and ChIP-seq data. So we did actually include this bulk reference in our package of Capybara so that people would not need to actually mine this reference by themselves. As well, if people already have prior knowledge in what reference they want to use and prior knowledge regarding which tissue are these cells from, they can completely ignore, just jump into the second step instead of following the first step. Here we would demonstrate still the first step, assuming that we don't really have any idea about what is the reference. And what is the cell type. So here we will first load our bulk reference from the Capybara package. So in the bulk reference, we have both the raw data as well as the RPKM in case people would like to normalize them using different method of their in bulk RNA sequencing data. I would suggest using the RPKM counts for the downstream analysis. Here we would showcase this classification using a data set that involves a mouse pancreatic isolate data set. So this data set is from Barron et al. And they profiled both mouse and human. So here we're using the mouse data set in this showcase. So the single cell atlas that we are using here is again the mouse cell atlas. We are using this as a background to classify the single cell RNA sequencing data of the mouse pancreatic cells. So we also include this data set in our package, so feel free to play around with this data set to see how it works in the pipeline. So this first two steps is really to get the data set from the package and then read the data set as the raw count matrix and reading in their labeled metadata. So to run quadratic programming using the bulk bulk transcriptome, we will just call the function to run a one-round quadratic programming analysis where we pass in the raw data of the bulk and the raw data of the Barron data set. So it involves the multiple different parameters involving the scale bulk SC. This means, do we want to scale the bulk down to a single cell level? And do we want to use Unix part is like, do we want to use Unix-based parallelization? How many cores would you like to use, and where do you want to save your files? So for Windows users, we would suggest changing the parallelization to force and set the cores to 1 so that it can run in serial. So further with this quadratic programming, Yeah.
Can I ask a quick question?
Yeah.
So what does a single round of QP analysis mean? So like returning to our discussion about the methods, yeah, what would a multiple QP analysis look like?
So a single round of QP analysis would involve one round of like, so that basically means we starting from the raw counts and then doing the normalization, doing the scaling, and then doing the logging, and then run the quadratic programming once. So it's basically a wrapped up version of like running the different steps of QP. It doesn't necessarily mean like running multiple rounds. So maybe it's a naming issue here. Yeah, sorry. So moving on, so here we are also loading our single cell reference data. So the single cell reference data is from the mouse cell atlas, and we are loading the single cell RNA, the mouse cell atlas reference metadata first. So this metadata include a total of 6 columns that has cell name, cluster ID, tissue, batch, cell barcode, and annotation. So these are what we will use for the high resolution reference construction. So in this step, we are not really using the cell type annotation here. We're more of using the tissue type annotation here. So here, because we also run through quadratic programming with the bulk transcriptome with the single cell RNA-seq reference data set. So we would apply QP tissue by tissue, cell by cell using the mouse cell atlas. So because mouse cell atlas is a very big data set, it includes over 40 different organs and tissues and cultures. So we didn't include the counts along with the package. We did further separate so we did construct this background quadratic programming matrix here. And we selected 90 cells from each tissue type and from the mouse cell atlas fractional identity matrix, and then included a subset of that in the package, which can be used for convenience. So this sounds very confusing, but let me explain quickly what we actually did here. So so in this case, we are running quadratic programming cell by cell, tissue by tissue. So what we are doing here is that we are running the each count file of the mouse cell atlas from each tissue against the bulk transcriptome of bulk transcriptome that we mined from bulk RNA sequencing. So we will save all these data files that are basically calculating the fractional identity score for each MCA cells against the bulk RNA sequencing data. And then from this, we will select 90 cells that we are going to construct like fractional identity background. So you don't necessarily have to do this step. You can use the entire QP matrix to serve as the QP background. So we get all of these QP scores for the annotated cells, and we did save this subset of the QP matrix in our package so that people can directly using this subset to see which tissue type their classification into in the MCA. So we separate the mouse cell atlas tissue into fetal and adult and saved these as embryonic background or fetal background inside of our package. So then the next step is to identify the tissue correlated in the reference to our sample single cell data set. So as I explained here, that we included the background inside of our package, and here is us trying to load the adult background into into R, and then we load the QP background here, and we also load the QP score of our sample, which is our pancreatic data set. And then we do perform the correlation calculation here. So we use the correlation calculation of WGCNA, which significantly improves the speed in calculating the correlation. And we here select a cutoff of the 90th quantile of the correlation matrix to perform a binarization. So this can be flexible depending on what people would consider as a high correlation. This can be, the cutoff can be modified based on prior knowledge and the structure of the correlation matrix. And then based on this binarized binarized matrix, we will be able to actually select the cells, select the tissues that actually capture at least over 100 cells inside of this single cell RNA sequencing data. So the major outcome of this bulk classification to the MCA tissue here involves stomach, pancreas, small intestine, and then muscle, and which starting from the muscle, it shares very small percentages it represents very small percentages of the cell population. So from here, we will select our tissue to include in this data set. So here we included 3 different major cell types in this bulk classification, including stomach, pancreas, and the small intestine. So we go into the second step and generate a high resolution reference for the continuous identity measure. So so the first step is to systematically construct a high resolution reference. So here, again, we are going through the counts from the mouse cell atlas, tissue by tissue, cell type by cell type, and to get all of the cell counts that are involved in these 3 tissue types. And then here it is, we perform some sort of a cleaning into the mouse cell atlas, because some of their labeling were exactly the same but cannot be identified as exactly the same. So so we perform some sort of a cleaning, and then we filter out the cell types with less than 30 cells, because in the construction part, we require we use a total of 90 cells to construct a pseudo bulk. However, we only require 30 cells minimum to represent the cell type, so that for cell types with between 30 cells and 90 cells, we will perform a resampling to compensate for the 90 cell reference cell number requirement. So, so here to construct a high resolution reference, we will call the function from the package, which is construct high resolution reference. And in this function, it will take in the count matrix, which is the mouse cell atlas count matrix that we are here to use to construct the reference, and the column data. So this is basically the metadata that we want to include to call the different cell types. And the criteria is a column that you want to construct the reference based on. So the reason why we include the criteria here is because in the mouse cell atlas, it has very detailed cell type distinction. So it involves a lot of cell types with a particular gene expressed, and using that detailed resolution reference versus we don't consider any marker expression will give us very different reference as well as the result-wise. So in this function, it also takes a number that say like how many cells would you like to use to construct the reference. So the default reference cell number is 90 cells. So you can feel free to change that if other numbers could work better, and if prior information and the structure of the cells and the reference look different. So based on the reference list, we will be able to call quadratic programming using this reference data frame that is, so I apologize that this line is incorrect. So this line is from older version of Capybara, and I forgot to update this one. So I will update this one later. So the reference data frame will be saved as a part of this reference list constructed from this construct high resolution reference. And using this, we will run the reference data set as described with the constructed reference to construct a background score, as well as from the query data set, we will also calculate a QP score for based on this constructed reference. And these two files will be saved in these file names. So based on these quadratic programming matrix, we will perform the empirical p-value calculation, which here we would load in the background matrix and the test matrix that involve the samples. And then because the matrix, the quadratic programming matrix will also include two columns involving the Lagrangian multiplier, suggesting how much we have pushed the calculation to the limit, as well as an error term in regarding to the linear combination and linear regression. And then here we would perform the empirical p-value, calculate the empirical p-value matrix for the reference RNA sequencing data set. And then as well as we will perform empirical p-value calculation for the sample data set. And using, by inputting these two into the binarization, it will be able to perform a Mann-Whitney test to further binarize this data set, binarize the data set so that we get a discrete classification of the cells to their particular cell type. So, so then we can further convert the binary form of the matrix to the discrete classification form, and in this classification data frame, it will include a column of barcode and a column of what is their classified call. So in the cells with multiple inferred identities will be marked with multiple identity, and cells with no significant inferred identity as unassigned. So we check the classification result against the manual annotation of the Barron et al. Dataset from 2016, and we were able to see that we were able to perform pretty well to map our prediction to their manual annotation. So in the, yeah, so in here we can see this diagonal here, and because in our reference data set, we don't have a detailed breakdown of all the different cell types here, so we are mapping them to endocrine cells, and I do believe they are all endocrine cells in this population. So here, so up to this point, I have demonstrated to you guys how Capybara actually run through to classify distinct cell types. And in the next part, I will be describing how are we going to analyze the cells with multiple identity. So is there any questions at this point? Okay, so here in this part, we are analyzing the cells with multiple identity. So we here I showcase another example with the cardiomyocyte reprogramming dataset that can be found under this GL number. So this dataset contains 6 time points of this reprogramming process between day -1 to day 14, where day -1 marks the day of transduction with the 3 transcription factor, and day 14 is where the cells were sorted based on the reporter gene. And the data set can be downloaded in the terminal as well as in R, which we provide the links to download. So here we pre-process the data with Seurat, so to give us the different the clustering and different information about the data quality. So to perform classification, again, we did a tissue level classification where we did find good mapping to neonatal heart, neonatal skin, and neonatal stomach and fetal lung. So in here, we included 4 major different tissue type, including the skin, stomach, and heart and lung. So we further constructed the high resolution reference and then performed the discrete cell type classification where the results can be found in the paper. And I will update this tutorial with those figures included. So then we will next filter the cells with multiple identities based on their QP scores. So a question that have come up in our mind is, how do we know if these multiple identity cells are actual multiple identity cells? Like, what about if they are false positives? So we consider an idea that because we know the QP score are measuring the fractional identity for each cell. So we only consider that the cells with like both identities have a significant QP score, which is greater than 0, which we define as greater than 10 to the -3. So if the QP score is greater than 10 to the 0.001, 10 to the -3, then if the QP score to both identities or multiple identities are greater than 0.001, then it will be considered as a true multiple identity. So this threshold can also be modified as an input parameter to kind of including more or be more stringent about how do we want to filter out these multiple identity cells. And then if one cell, the multiple identities of the cell, a couple is very close to 0 while one of them is very significantly larger, then we will reclassify that cell as single identity instead of keeping them as multiple identity. So in that sense, we will filter our multiple identity cells by calling this function, and then we will be able to get the actual multiple identity and have a revised version of the classification. So using the multiple identity cells, we will be able to calculate the transition scores of the different systems. So here we will calculate the transition scores in this cardiac reprogramming process. So it is a very simple function that you just call transition score, and input the multiple identity list that includes the cells and their multiple identity labels. So here this is the result that we obtained for this direct cardiac reprogramming process, where we see there is a high transition score in day 1 and day 2, and starting to decrease at day 3, which this result reflected is also mirroring what they have found in the paper in Stone et al., where the cell fate determination is starting early within the first 24 to 48 hours in the reprogramming protocol. So that's all for the tutorial of running Capybara. So, yay.
Thanks, Wenjin. So I had a question about the bulk classification on the Stone data. So it comes up with neonatal stomach. Like, how should that be interpreted or handled? Because then obviously when you move to the high resolution classification, we don't see stomach again after that.
So, so the reason why the stomach come up, I think it's because, so first of all, in the reference, we don't actually have a cardiac fibroblast population labeled inside of mouse cell atlas. So it's basically trying to find that fibroblast population everywhere else in the tissue list. I don't think there is particularly a biological interpretation to this, because so the tissue level classification is supposed to be broad, and so that we can include, we can be sensitive enough to include as many cell types as we need for the downstream analysis. However, the high resolution should actually be more sensitive in actually mapping into the different cell types in that sense. So,
so you're saying don't read too much into that initial bulk classification.
Yes, the bulk classification probably requires some improvement along the line. We will continuously work on that, and I have a couple ideas, and we will work on that.
Yeah, and obviously the point to raise here is that if you have the ideal single cell atlas, so in the Capybara preprint, you've shown this with motor neuron conversion to motor neurons. So having that spinal cord developmental atlas has been really valuable in that context. And so it's my expectation, I think it's our expectation, that as the database of single cell atlases expands, then this approach will become more powerful.
Yes, and if people do have prior knowledge, then the tissue level classification shouldn't be necessary in general. So I would definitely suggest, if there are prior knowledge involved, then maybe ignore the tissue level classification, because it can be disturbing sometimes if you get a completely unexpected tissue type coming up. Like we get ESCs in our IEP reprogramming protocol. However, it kind of goes away. It goes away in the high resolution classification. So it was very disturbing for a while that why did it come up, and however, it did go away. So yeah, we shouldn't interpret too much into this. That's all.
So then on the flip side of this, if you have your custom reference, you have prior knowledge, but that prior knowledge is very biased and blinkered. So you think you have a good reference to compare your data. So say with the IEPs, where say we were convinced that they resembled some kind of kidney cell type. So we used a kidney atlas to compare these reprogrammed cells to. Then what are the dangers of selecting an inappropriate atlas, would they all be unassigned in their identity, or would we have a lot of false positives?
So I think what would happen is we will have false positives, definitely, and you will get a lot of unassigned cells, as well as further looking into the quadratic programming result, they will be having a very high error. So like they are not mapping very well to any cell types. Technically, they should all go into unassigned. However, there is definitely because of the similarity in some of the gene profile, they could be classified into like forced classified into a couple like different cell types. And in that scenario, we could definitely evaluate the metric inside of quadratic programming to see if the errors are way too big, or the Lagrangian multiplier is just like way off the board. So that would definitely.
I forgot about that aspect of QP, so that could be really helpful in this context if we select a bunch of inappropriate references and look at those metrics.
Yep. So, any other questions? Okay, thank you.
Thank you, Wenjin. That was great.